The overall goal of this procedure is to confine cell-free protein synthesis reactions in cellular relevant volumes. This is accomplished by first fabricating a PDMS device using layered soft lithography techniques. Then, a cell-free protein expression reaction is mixed, introduced into the device, and an ensemble of isolated reactions are formed within an array of microfabricated chambers. Next, the device is imaged using time-lapse fluorescent microscopy. The final step is measuring the fluorescence intensity of each well over time. Ultimately, these fluorescence traces are used to show protein expression noise in confined cell-free reactions.
The main advantage of this technique over existing methods such as studying noise in live cells is that synthetic systems may be easily probed for conditions such as confinement, which can't be easily controlled in living systems.
To begin the microfabrication process, first make a 20:1 weight ratio of PDMS mixture by measuring 10 grams of PDMS base and 0.5 grams of curing agent in a small plastic dish. In another dish, weigh a 5:1 PDMS mixture for an amount enough to fill a control valve master mold up to 1 centimeter thick. Mix both parts evenly with a spatula, and place the dishes inside a vacuum desiccator for 20 to 30 minutes to degas all bubbles from the mixture. While degassing, place the control valve master mold into a glass container. Pour the degassed 5:1 PDMS mixture onto the control valve mold. Transfer the glass container into the vacuum desiccator and degas.